How do you do? My name's Phil Coelho. Thermogenesis is a division of Cesca, but this is a little different presentation than you might have been anticipated, but since CAR T cells are so hot now, I thought that this might be of interest to the crowd. The semiconductor industry has the miracle of, as their production goes up, their costs go down and their quality goes up. If the people manufacturing CAR T cells don't figure out how to follow that pattern, then either the country is going to go bankrupt or many patients are in fact not going to be treated with these pretty wonderful therapies. And so our company has some technology we feel which may in some way assist that effort to manufacture these complicated cell populations a little less expensively. As you can see from the, these are devices. This is the actual production schedule for CAR T cells. It has quite a few steps. You begin with a donor blood or leukapheresis. Then you isolate mononuclear cells, you select CD3 cells typically, then you genetically modify and expand them. There's a lot of washing steps throughout it. At the end of the day, you have a final formulation, and then you typically will freeze it at the end. And all of these things are complicated. We have some devices that may make it a little simpler. They certainly will automate it. The device is actually a reusable, or rather a not usable, single-use plastic cartridge of some complexity shape. It has a control module and a docking station. This is a cross-section that you'll see it's basically a conical shape. And it also has two additional compartments where depleted populations go and harvested populations go. And it has a control module, can operate that because it does everything that it does while it's spinning in a centrifuge. And that's a little unusual. This is a standard centrifuge that Thermo Fisher sells and is modified for our company, markets them worldwide. They call it thermogenesis compliant. It operates like many of these things on the fact that these cells have different densities, so when you spin them, they wind up in different locations than when they started, and that works to our advantage in terms of isolating cells. So when you drop it in, if we took a cross-section vertical to the floor, you'd see the blood or leukapheresis in down to the cone. The control module allows cell fractions to move to different locations. This is a cross-section now. Let's say that it's spinning. It's a swim bucket. So this is a cross-section that's now parallel to the floor. So you see that the red cells have come down. Then you have all your white cells right here. You have your platelets and then the plasma. And they line up according to density. Now, these little yellow dots are actually simply locations where the optical sensing interrogates the cartridge. The centrifuge then slows down and we passage all these cells we don't want over to another cartridge. And then even the white cell fraction lines up according to density. So we have the ability when we harvest to just simply slice out the fraction that's important. So for instance, the single most important area is at the start of CAR T cell. Because whatever cells you get from the patient, that's it. Things don't get better. Basically, what you lose off the original collection are cells that are simply not going to be around to expand or do anything else. And this is the way that most people are doing it. It appears to be something out of the 19th century. It's a manual process. It has a whole series of steps. I've redlined some simply because they're alerts that things are slow. Things are toxic, things have to be washed, and it's an open system, which means it's done under a Class 100 hood, which itself is in a clean room. And the recovery, I'll back up, is basically pretty terrible. You've lost half your cells right off the bat. Our strategy is in a closed system. in a fraction of the time, basically to go ahead and remove the red cells, remove most of the granulocytes, and then rotate it and resuspend the platelets, and then go ahead and haul the mononuclear cells into the harvest compartment. You get a GMP compliant readout at the end of it, and you can see that we've captured 94% or thereabouts of the mononuclear cells and depleted all the other parts which now are considered to be contaminants. The other part of that I'm showing is the X-back system, which is a new way to select cells other than magnetic beads. So this uses tiny microbubbles that are about a third or a fourth the size of the cells themselves. We attach them with antibodies, and so what happens is once we attach them, we incubate them. Then we can go ahead and spin, and when the cells that we don't want go down, we can then deplete them. The cells that we do want have actually floated now, and then we can break the linkage with the microbubble and bring them down and harvest them. That's a relatively fast process. This is a flow cytometry chart that shows the key element here, which is that these cells, CD3 cells, are recovered at a very high percentage, and the key monocyte, which you do not want there, is depleted at a very high percentage, even though these cells are quite similar density. So this is basically a home run here. The time frame to do this is described from start to finish. It's about 2 hours. This is a comparison with the competition in terms of a time frame. It's considerably faster. Cells that you don't get at the start, again, don't come back. And so when you begin your expansion process, your induction expansion process, now you have considerably higher recovery than the state-of-the-art today. The bioarchive system is right at the end. Now, this was a robotic cryogenic system, originally developed for cord blood, which is an interesting model because it utilizes, or rather it's attempting to protect both the stem and progenitor cells, hematopoietic and stem and progenitor cells, but also the active complement of T cells. I won't go into too much detail, but it attempts to do the freezing process, cryoprotection freezing process, as perfectly as possible. It, again, has the identity tracking throughout it. It does the controlled rate freezing, provides a curve, so you know every one of them is basically perfect. They're done unit by unit, and in CAR-T, it's all single units. So the N is 1. And the net result of it is, in the cord blood field, pretty remarkable. If you know how to lead a Kaplan-Meier curve, basically as soon as you use the bioarchive, you have accelerated myeloid engraftment, and then you have accelerated platelet engraftment. And these are significant numbers of patients now, and it's highly significant, the accelerated engraftment. And as you would expect, if you engraft faster, you survive more, or more people do survive. So their motility is decreased, and their survival is increased. So it becomes a risk factor, not different than HLA or cell dose. So we didn't cover all the manufacturing process because it's a 15-minute process. All this data was performed by Dr. Pablo Rubenstein, the father of all cord blood banking. We have begun to automate. In this cartridge, we can elevate the temperature, move it where we want. We can mix inside of it. We developed a device that goes ahead. This data has come as a result of doing it. It can both mix and heat and do everything else we want. And we're now in the final design work that looks at a fully automated device where all the reagents and what all go into the system as well. And when you compare this in terms of scaling out, you now can do this in batches of 1. This is the main competition, a wonderful device by Melteni. It's about 160,000. This is considerably less. And the net result of that is each technician can be considerably more productive in a work cycle to generate these cells. So it's a closed system, and we believe will allow scaling out of the production of these CAR T cells. And that's it. Thank you.